Life as we know it takes many forms, but at the microscopic level, it all boils down to two basic types of cells, prokaryotic and eukaryotic. Prokaryotic cells are simple and small, like tiny studios. Bacteria and archaea belong to this category. They hold the blueprints of life, their DNA, directly within their jelly-like interior. Eukaryotic cells, on the other hand, are more like complex mansions. They are larger and more organized, with a nucleus that acts as a central command center, housing their DNA. Animals, plants, fungi, and even single-celled organisms like amoebas are all eukaryotes. The difference between these two cell types might seem small, but it's actually a giant leap in complexity. Understanding how eukaryotic cells arose from their simpler predecessors is like uncovering the origins of complex life itself. The evolution of eukaryotic cells is a captivating mystery. How did these intricate structures arise from the simpler prokaryotic cells? The answer, it turns out, might lie in a groundbreaking idea known as the endosymbiont theory. Imagine a single-celled organism, a prokaryote, going about its daily life, perhaps absorbing nutrients from its surroundings. Now imagine this organism engulfing another prokaryote, but instead of digesting it, something extraordinary happens. The engulfed cell survives and continues to thrive within its host. Over time, this unlikely partnership becomes mutually beneficial, with each cell depending on the other for survival. This, in essence, is the core of the endosymbiont theory. The endosymbiont theory, championed by the visionary scientist Lynn Margulis, proposes that eukaryotic cells evolved through a series of symbiotic relationships between different prokaryotic cells. One prokaryote engulfed another, and instead of becoming lunch, the engulfed cell took up residence, eventually becoming an integral part of its host. This process, repeated over millions of years, led to the development of organelles, the specialized structures within eukaryotic cells, mitochondria, the powerhouses of the cell, and chloroplasts, the photosynthetic factories found in plants and algae, are believed to have originated from such ancient symbiotic events. The endosymbiont theory revolutionized our understanding of cell evolution. It painted a picture of life not just as a struggle for survival, but also as a dance of cooperation and interdependence. The evidence for the endosymbiont theory is abundant and compelling. Both mitochondria and chloroplasts possess their own DNA, distinct from the DNA found in the cell's nucleus. This DNA is circular, similar to the DNA found in prokaryotes, and suggests a separate evolutionary history. Furthermore, these organelles divide independently of the cell itself, replicating in a manner akin to their prokaryotic ancestors. They even have their own ribosomes, the protein-making machinery of the cell, which are more similar in size and structure to those found in prokaryotes. The work of Carl Woese, who pioneered the use of ribosomal RNA to study evolutionary relationships, provided further support for the endosymbiont theory. His research revealed a deep connection between mitochondria and chloroplasts and their prokaryotic ancestors. The story of eukaryotic cell evolution is a testament to the power of collaboration. It reminds us that life's grand tapestry is woven from threads of interdependence, with even the most complex structures arising from the harmonious interplay of simpler parts.